Welcome, everyone. I am Adrian Lee, the Pittsburgh Foundation Chair and Director of the Institute for Precision Medicine at Pitt and UPMC, and I'm Professor in the UPMC Hillman Cancer Center. I am privileged and really excited today to have a fireside chat with guests who I'm sure all of you know, Drs. Tureci and Sahin. Dr. Tureci is the Chief Medical Officer at BioNTech, and Dr. Sahin is the Chief Executive Officer. Welcome.
Thank you, Adrian.
So I think by now, everyone in the audience has heard your background, but for the up-and-coming scientists who are in the audience, who are both in the lab or they're maybe entrepreneurs, I wonder whether you could just talk about your background, how you met in your career, and how you ended up in this role at BioNTech.
So I can speak for the two of us here. We are both, Adrian, we are both physicians by training and immunologists by passion, and out of desperation became entrepreneurs. The short version of the story is that we experienced as young doctors on oncology wards that there are not many standard of care medicines in our armamentarium when it comes to treating patients with advanced solid cancers, for example. And on the other hand, because we started very early to do research, we were very much aware of the potential of technology and science. And the question for us was, why can't we get those potential treatments faster to patients? And we learned after a while that we have to take over the responsibility for developing our science to medicines. And that was the reason why we started to found companies, and BioNTech is the second one.
Yeah, no, that's a great story. And you mentioned this word passion, your passion for this. And I heard Dr. Sahin in a TED talk about this. There are many people out there that are now couples in science. Actually, my wife is from Germany. She's from Berlin, and she's actually a scientist as well. We're a scientist couple. And we, funnily enough, say that we are passionate about, we live our life is science, yeah, at home, and our kids say that as well. Maybe you can expand on that, on how you handle this work-life integration, how you have this incredible kind of handling of, you know, doing the work and then living your life as well.
Yeah, I think the normal idea is, of course, separating work and family. And we have here the privilege, being scientists, to do what we love, yeah, and we don't need to separate that. So we are a scientist family, yeah. So we talk about science at breakfast, yeah, during lunch, and if we have sometimes dinner, also at dinner, yeah, and we live that, yeah. And it is like sometimes it feels like you have beautiful flowers. Yeah, you have all these experiments and you want to know what is coming out of the experiments, how the flower is going. It is really, really increasing and providing richness in our life. And I see it as an extreme privilege to live this type of life. And of course, connect that to everything what also matters, family and time for doing other things. But we don't need to separate that in a fashion. This is work and this is private life. We just mix it up.
And a major feature of PMWC, as you know, is to have companies there, and there'll be a lot of entrepreneurs in the audience. So I wonder, and you had said this about controlling the science. So does that, having the company, allow you to control the destiny more than you had in academia, or how is that different to when you were academic physicians versus you now being in the company? You're controlling the direction more?
You know, Adrian, we try to combine that in our company. We are still scientists in our heart, and we believe that also pharmaceutical development needs to be deeply rooted in understanding the scientific mechanisms and understanding the science behind the technologies. So there is no firewall between basic and mechanistic science and developing innovative medicines and application of the science in that regard. So what we try to do in our company is preserving this academic and scholastic spirit and the curiosity, and at the same time, for those programs where we see a clear clinical translation, setting up the very focused and forward integrating way as pharma normally works. So we really think the mixture is the secret of success here.
Yeah, so maybe I can just ask you that a bit deeper, because you said in your acceptance speech last night that the success of precision medicine is upon kind of what you just said. You need to understand the basic principles of immunology, how vaccines work, cancer, before you can then apply them. But obviously, and there's a great deal of at the moment of the push to just translational research, and basic science is somewhat struggling. And in the US, it's struggling to get funded. I don't know how that is in Europe. But so in the company, do you focus on some basic science, or you interact with academia to collaborate on that?
Yeah, so I believe there are two misconceptions. Yeah, the first misconception is the idea that the direction goes always from basic science to applied science to product development, and this is not true. Actually, in a real situation, if we encounter a challenge in the development, yeah, then we want to understand what are the basics behind the problem. Understanding the problem is always the key in coming up with a solution. Of course, sometimes you can come up with a solution by just a discovery, yeah? But if you really do translation, and if you struggle, for example, that the potency of a vaccine is not sufficient, you have to go back and understand what is the challenge. And then you end up, for example, with immunology. And it's not just simple immunology. You have to understand which cell types, for example, take up your vaccine. What are the mechanisms behind that? How can you ensure that the cells translate that? It is biology, yeah? And this makes it wonderful. So it is, at the end of the day, you are dealing in a company like in ours with basic science, applied science, product development at the same time, yeah, and that makes it beautiful.
Yeah, no, that sounds super exciting. Yeah, I know these sometimes get compartmentalized, as you said. You know, you have production over here and basic science over there, and I think it's, you know, I think when people look at this last period of time, they see this amazing, you know, innovation. And maybe I can ask you about this. So the 18 months, obviously, for the two of you must have been, you know, an incredible kind of storm of events. If you step back like to 2 years ago, let's say 6 months, say this is now like 6 months before the pandemic started, did you think you were on the precipice of doing something as monumental as this? Or you were like all scientists just hoping that the next experiment was going to be the one to do something? Or did you really have an idea, yeah, I really think our technology is now at that point of a major translation and a major impact?
Yes, so we were preparing end of 2019. We were preparing for ourselves for important milestones. Our cancer vaccines were transitioning into the proof of concept phase, which means big randomized trials, comparison to a standard of care, which needed to be conducted internationally in different countries. So that was already a big step and an important important step, and a step which not quite in 2020, but thereafter would then bring a proof of concept for our innovative treatments for the first time. So we expected that 2020 and 2021 would become important years for us, but not at that scale, because COVID-19 was not at the horizon. And in fact, when the pandemic started, all those best laid plans were shattered. And what we did was, if life gives you lemons, in this case, a virus, we made a COVID-19 vaccine out of it, and which meant we invested all our force and our teams, which were slowed down by the pandemic in their cancer vaccine activities. We redirected that towards the COVID-19 vaccine development.
Yeah, it sounds as like you took lemons and you made champagne out of it, yeah?
That's a trick, yeah. Yeah,
So,
yeah.
Go on.
Yeah, just to add, actually see on the way of proving something in a efficacy trial, yeah. You have, of course, other trials before, and we had very successful early clinical trials where we could show that our cancer vaccines really induce very strong immune responses. We have observed objective responses in cancer patients, even in advanced cancer patients. So we knew that we had a powerful therapeutic platform, but the proof in cancer immunotherapy takes much longer than developing a vaccine during a pandemic.
Yeah, so maybe I can come back to that, because I'm sure now mRNA therapeutics and vaccines is part of our dictionary, but it's happened like pretty quick. Obviously, I knew about mRNAs as treatments many years ago, like many people, and there were challenges. But suddenly this has become so, will become just pretty much dogma, yeah? And so what made you start with them as opposed to proteins or small molecules? What made you initially think that mRNA was going to be the useful drug, let's say?
Yeah. Do you want, or
yes, I can start. So actually, Adrian, we tried all those. This was in the 1990s, where cancer vaccines were starting to become a thing. Yeah, and as others, we tried peptides and proteins and all the other vaccine formats, and at some point also mRNA, and we understood that mRNA has a couple of really interesting features: intrinsic adjuvanticity, the precision with which you can really represent the target. It had also features which were disadvantages, and therefore it was overlooked by many, namely that it was short-lived outside cells and inside cells, and translational efficiency and therefore potency were not great. So we understood that the advantages were sort of bigger as the disadvantages and hurdles, and we saw a path forward to improve on those disadvantages. And so that was the epiphany end of the 1990s, and we started to optimize mRNA. Since then, we optimized it on different levels, including the key disadvantage, namely potency and short-livedness in cells, and could thereby make it to a tool which exactly addresses what we need for cancer vaccines. In particular, also personalized vaccines. The manufacturing part is also an important advantage. It's synthetic, it's fast, much faster than recombinant protein production, and that's important for personalized approaches.
Yeah, just to add to that, so I believe in the 90s there was a hype technology. These were DNA vaccines, yeah, and we started also with DNA vaccines, but understood that DNA vaccines have a fundamental problem. So it was not possible to bring them, to transfect them into dendritic cells, yeah, human dendritic cells. And when we made our first experiment in end of the 90s with mRNA, we got 80% transfection rate in dendritic cells, and this was a clear evidence, and that the fundamental problem to resolve for vaccine development can be addressed with a mRNA vaccine, and that all other aspects are based on technological challenges, and with time we might be able to come up with solutions.
You know, it seems to me it's kind of, and I've heard others say this, a little bit of kind of a science fiction story, and I'll truly admit I don't really understand how this works. So you inject an RNA in, be it in a lipid or not, it gets taken up by cells, and then it's what, grabbed by ribosomes and then translated, and that's it. And does it have to be adapted for that mRNA translation, that presumably there are parts of it that make it translated better? That's all of these things that you've spent 20 years working on to maximize?
Yes, yeah. Yes, exactly. One example is the synthetic mRNA has the same structure or anatomy as mature processed cellular mRNA. So you need the cap, you need the 5' and 3' UTR and the poly-R tail. And one of those work streams on which we have worked is to optimize the cap region, the UTRs, the way the poly-R tail is constructed, in order to ensure that this mRNA, once it has reached the cytoplasm of cells, is translated with priority, basically. That it stays long at the ribosomes, that the translational efficiency of how much protein is generated is increased, and this is one of the important parts to ensure this VIP position at the ribosome, and not any ribosome. That's also an important aspect, to understand in which cells you want to see that. As an immunologist, you want in particular lymph tissue resident dendritic cells to do the job. That means all those screenings to find the right components, the right cap, the right UTR, had to happen in human dendritic cells, which had the right phenotype. Yeah, so this is one piece of the puzzle, and there are more.
Yes, like you said, it's the basic science. Without the basic science of understanding, mRNA translation through ribosomes, which is pretty complex, yeah, and now you have all these, because you have a new era of mRNAs, you have these pRNAs, non-coding RNAs. Presumably you're only focused on translatable RNAs, or do you have interest in this other era of RNA biology? Circular RNAs, for example, you don't consider any of these other RNAs, yeah?
There will be plenty of mRNA applications. So at the end of the day, the mRNA toolbox that we have generated does not only include normal in vitro transcribed mRNA, but it includes also self-amplifying mRNA. We developed a technology which we call trans-amplifying mRNA. That means we co-deliver a replicase and a target mRNA which is amplified by the replicates without the replicates being amplified. And as you said, there are other RNA, non-mRNA based applications, microRNA, and we are just learning about the PV sequences. And the other parts of the solution, that means, for example, the delivery tools, can still be used, yeah, to work also with other type of RNAs. So that means once we have a toolbox, we can ask which part of the toolbox could be repurposed, yeah, to enable the development of other type of biomolecules. But what is also important is, for example, what we learned is when we modified the 3' UTRs, yeah, that it was important to evaluate that this 3' UTRs are not targets of microRNA. It's at the end of the day, we know that also synthetic mRNA, as Ozlem said, will behave like normal mRNA and is therefore prone for regulation by cellular machines, yeah, including microRNA.
So I would like to come back to the question of delivery in a second, but maybe I can just ask you one quick question. Is there a size limit to what you can express? Because obviously that would be important if you want to express insulin or something. What's the size limit for the mRNA that you can encode?
In principle, not. So at the end of the day, the technological challenge is just to create a template which is long enough. So we have examples of even getting 20 kilobases, yeah, into cells, yeah. Wow. And you have just to ensure that the longer the molecule is, yeah, the more impurities you might get. But technically, everything what you can include, what you can package into a lipid nanoparticle or other tool for delivery can be delivered to cells.
Yeah, that's great. That's great. So I want to come to that question of delivery. So actually, after this fireside chat, we have a panel session talking about disruptive technologies, of which obviously mRNAs are part of that. And actually, one of the speakers is Catherine Whitehead. She is at Carnegie Mellon University and is an expert in lipid nanoparticles for mRNA delivery. She actually just gave a TED talk a couple of weeks ago. I highly recommend you look at it. It's really impressive how she talks about these, you know, the amount of engineering you can do on these lipid nanoparticles now to get this. And I wondered whether you could speak to this, what you've done and what you envision for, like you had said, targeting to the right cell, making sure it's delivered, make sure it's unpackaged correctly. I mean, is that got a long way to go, or you think that's relatively mature now, or you think biodelivery is still a major area for advances?
Yeah, maybe I start with them and then you can add. So again, delivery is not just a single set of challenges. So if we talk about delivery, we have to ask the question, which type of delivery? What is the injection regions? So we started with our lipid nanoparticle or lipid lipoplex work in 2010, yeah, about more than 20 years later than the first publications, preclinical publications came out for lipid-based delivery. When we started, we were interested to deliver our mRNA exactly in the cell type that we discussed into dendritic cells. And the problem was that we could deliver mRNA into mice, yeah, but it was just transfecting all kinds of cells, but not the ones that we wanted. So we figured out and found that certain particles at with certain physicochemical properties and size could be delivered in a way that that the nanoparticles did not went to the liver, did not went to the lungs, but went to the lymphoid organs, to the spleen and to lymph nodes, which was our intravenous dendritic cell delivery mechanism. And this is part of our cancer vaccine program. And it's also part of our CAR T cell vaccine program, and it's also part of our program using this type of delivery to deliver non-immunogenic mRNA to induce regulatory T cells, for example, for treatment of multiple sclerosis. Yeah? So the solution works, but when we were encountered with infectious disease vaccines, intravenous delivery is not suitable. Yeah? And the type of particles that we had developed for the cancer treatment were not suitable. Here, the way to go is, for example, using lipid nanoparticles, yeah, or polyplexes, yeah. So, for example, polyethylene imine-based particles. And again, the lipid nanoparticles are not just one group of particles. There's a lot of modifications how you build that, yeah, and how to engineer that. And it also depends on the lipids. It depends on the groups that you use for functionalizations. For example, for functionalization, you can use PEG, yeah, or we have published that you can use polysarcosine or other groups. So that means it's again really a huge set of elements that you can use to screen nanoparticles which do a certain job. In our case, it was again intramuscular injection and bringing the mRNA to dendritic cells in the lymph node, for example, in the regional lymph node. We believe that the challenge for IM and intravenous delivery of such particles into dendritic cells have been solved. Of course, this can be improved. But if you would ask, did you solve the delivery to human brain? Did you solve the delivery, for example, to use mRNA to deliver, to use nanoparticles to deliver into muscle cells, but not just to the injection site, but to all muscle cells? The answer is no. Yeah, and I would say we even don't have an idea how to do it in a way. Yeah, so that means it is not only a technological solution. Again, it's a biological solution. Yeah, we have to understand what are the biological barriers to overcome to reach the cell types in a way that we can, for example, deliver mRNA to brain cells, to neurons, yeah, or to all type of muscle cells. Yeah, so this is an unsolved challenge, and maybe in 3, 5, 10 years, the industry comes up with solutions.
Ozlem, do you want to add to that?
No. I think Ugur has covered all. It's delivery with all sorts of particles and lipoplex and so on is a field of very high potential. So we will see innovation and breakthroughs there. As Ugur has said, some of those concrete questions or needs have been addressed, but there are so many others. So it's a very interesting field.
Yeah. So maybe I can dig a bit deeper into that then, because Ugur, you beat me to I was going to ask you about the cancer research paper, and but moving on. And I think the important point about the cancer research paper to me, which was in 2010, just shows, and I think, Ozlem, you'd said this before, how much basic science has happened over time. It's not like, I think the public probably see this as, oh, they decided to make a vaccine and in a year they made a vaccine and this is incredible, but it's based upon 20 years of basic research and preclinical research. So I think that's an important point to get across, that people don't believe this just happened overnight. It was an incredible amount of information that led to this. But how about at the meeting, at the PMWC meeting, we had a session on neonatal screening by sequencing, which is becoming very powerful now. So you can sequence by whole genome sequence infants in the NICU, and you can rapidly, within a couple of days, identify the genetic reason for their disease of interest. And obviously, many of those we can offer therapies, and many of them, probably just as many, we can't do much about it. And so then you think, wow, now we have this this other alternative approach to altering the trajectory for these cases. And people have proposed mRNA delivery for various of these diseases. But that comes back again to this delivery question, the long term. You know, you mentioned PEG, but people develop antibodies to PEG, for example, yeah? So what are you in BioNTech thinking about that, this long-term delivery for other, I know you focus a lot on cancer, and obviously you've done a lot on vaccines, but do you think about rare genetic disorders and other areas, or you are steering clear of that?
Yes, we are thinking about that. And you indicated something very important, at least implicitly. So there are applications where you need to deliver the mRNA a few times to solve the problem. Yeah. and even for cancer vaccines, we are now able, so we have patients who are under treatment with therapeutic cancer vaccines for almost 2 years. So repeated vaccination with cancer vaccines, even with the intravenous cancer vaccines, is possible. But in the particles that we developed for the cancer vaccines, we explicitly did not take in PEG because
Okay.
because we knew, of course, about the risk that with immunization with PEG there could be antibodies, and if we are really interested in an application where we want to deliver a particle or a protein in a non-immunogenic fashion, and it is required to do it not just for 4 weeks, 6 weeks, but it might be required to do it for a year. We have to much better understand the immunogenicity problem. And so that means what does it take to make delivery tools and messenger RNAs that avoid immunogenicity or limiting of the bioactivity of the molecule after 3 or 6 months due to lack of bioavailability, or even worse, due to allergic reactions. We are not yet there. So we need to address that, and again, the way to deal with that is to understand the problem. We do not clearly understand the problem of PEG immunogenicity. So it is known that antibodies could be generated, but we do not really understand what are the risk factors. We have an understanding how to avoid strong PEG responses. For example, some of the nanoparticles are designed that the PEG is disappearing within a short time, so that the nanoparticle becomes naked. So there are solutions, there are technological potential solutions, and we need to increase the space of potential solutions. And one option is to use other groups, which are not based on sugars. And so it's an exciting field. And we know that the immunogenicity problem can be overcome with, for example, with the use of antibodies. We should not forget that the early phase of monoclonal antibodies were limited just by HAMAs or antibodies, anti-drug antibodies. And now we have diseases where we can apply monoclonal antibodies for years. And we will come there also with mRNA therapies.
Yeah, and Ozlem, I want to come back to a comment you made at the when you accepted your award last night. You had said that you didn't do this alone. Obviously, like you said previously, this is built upon both your work and others' work. I'm in my lab, I do breast cancer biology. We do a lot of genomics, and I'm a real evangelist for sharing data. I think that's really enabled science. And I think the COVID pandemic is an amazing example of the sharing. I presume you were pretty much dependent upon the sharing of sequences for the spike region for your success. Yeah, you would say you were pretty much dependent. There are some dependencies. Is that true to say?
Yes, absolutely. And this is the nature of innovation. It does innovation, and in particular breakthrough innovation is not based on one single finding of what someone has had in their little room in their lab. It's really about having access to data, which not only past data, but the most current data. And this was amazing during the pandemic, that we had these preprint servers, and data was available instantaneously, and even basically at the fringe, at the bleeding edge of the SARS-CoV-2 research. That's absolutely essential for being able to make breakthroughs.
Yeah. So we have a session on that discussing about data sharing, federated learning, all of these things. And like you said, preprints have been a good and a bad thing. Yeah, it's been an amazing way to share things immediately, but some of the things got shared that perhaps shouldn't have been. Is there things which you would like to improve upon sharing? Do you often fret about, oh, I wish we had this, it could be shared easier, or you think that's going well for you, the ability to share data, etc.? I know this is difficult in companies where you have intellectual property and stuff.
Yeah, so I am really in favor of sharing data as soon as possible. So if you go to a preprint server, the first thing you see is the disclosure that this paper had not been reviewed. Yeah?
That only happened for SARS-CoV-2. SARS-CoV-2 caused that yes, disclaimer.
Yes, it is really important to have this disclaimer. And if you, the one wonderful example that I had seen in the early days of SARS-CoV-2, there was a paper claiming that smokers, due to a lower ACE2, had a better risk profile. Yeah? And it was not pre-reviewed, but while this paper was published, a few days later, several other papers came out and said, we can't confirm that. Yeah? So that means it was great. It was not the right information, but it inspired people to just check that and validate or disvalidate that. That's what I love. It is, at the end of the day, we live in a world where a certain piece of information can be rapidly validated or disvalidated. Yeah? The scientific community does the cleaning, yeah? And we don't need to fight, to have this fight with reviewers for years, whether a paper should be published or not published. It should be published, I think. Information should be published. And even with patents, also our IP managers are used now to make patent applications in light speed. So that means there is no reason, to delay publications of papers. So I really like that.
I'd like to ask you, I went to your website, and the website speaks a lot about cancer and not a lot about vaccines. And so I would like to ask you, what's exciting what's keeping you up about cancer at the moment? I know you've probably spent the last, as you said, you've turned around the company, and for the last 18 months you've lived vaccines, but presumably you'll go back to doing cancer. And I think what do you think is cutting edge? What do you think you'll be able to achieve with cancer vaccines for prevention and for treatment?
Ozlem, do you want to take that?
Yeah, so with our cancer vaccines, we are now in phase 2 trials, so the proof of concept trials, and we have two colors of cancer vaccines. On the one hand, we have off-the-shelf vaccines based on tumor-associated antigens, where every patient with a given tumor type gets the same vaccine. On the other hand, we have our truly individualized approaches, as I pointed out yesterday night in my acceptance speech, where each and every patient gets their personal and unique composition of their cancer mutation-based vaccine. With both approaches, we are now in randomized trials, and the data which we will obtain in those trials, which compare against standard of care, would be then the proof of concept. And we have to wait for this proof of concept. We have seen in phase 1-2 trials that our cancer vaccines are active, that we have objective response rates, and the proof of concept studies will tell us more. We are developing vaccines for treatment of established metastatic disease. And we are also developing vaccines for prevention of relapses, which means in the adjuvant situation. So the first treatment has healed the patient, but we know that there is a risk of relapse, and the vaccine is supposed to delay or prevent this relapse.
So this has been a fascinating chat. We're into the last 5 minutes. And what I would really love to do is for you to take off your lab coats and put on your business hats and ask you a couple of business questions, which may not be the life you're constantly living, but I know with Ugur being CEO, he must be thinking about this, and I'm sure, Ozlem, you are as well. So first, very quick question. Do you sense, because there'll be a lot of business companies, industries, and entrepreneurs in the audience, do you get a sense that they're more excited about mRNAs, like a year and a half down into the pandemic, that investments are going to increase in this space, particularly in cancer vaccines, for example, that there'll be more excitement coming from them?
Yes, of course.
That wasn't obvious. That was an easy softball question. So then the hard question. To come back, I would like to end on that point you just said, Ozlem, about personalized therapy. So I work in breast cancer, and my analogy, which is similar to what I think I heard Ugur mention previously, was that every tumor is different. The way I say that is that, you know, every tumor is like a fingerprint. No two fingerprints are the same. And this is why it's such a challenge, you know, at the level of genomics, at the level of the RNA, at the level of the methylation, at the proteome. It's just such tremendous heterogeneity. It's amazing that anything works outside of, you know, chemotherapy. Yeah. So if this is true and you want to give a unique therapy to every single person, how do you think business-wise this is going to happen? Because we have examples of this with, for example, CAR T therapy, and that's problem monetary-wise. How's this going to work in terms of the business side, the selling these? How's the healthcare system going to handle this cost of personalized, truly personalized therapy?
We are confident that this is not challenging. So the problem with cell therapy is that you really at the moment need the cells of the patient. They had to be transported, and then you have to connect that to this expensive out of the body culture and retransportation issue. So the future, if we really assume that a personalized cancer vaccine is approved, is what you would need is a next generation sequence of the tumor, yeah? Which is, we believe, part of future healthcare anyway. Yeah? What would be needed is just upload of the sequence, yeah? And we would have algorithms, and the algorithms would just design the vaccine, and then the printing of the oligos and the mRNA synthesis would start in an automated fashion, yeah? And just looking into a future, maybe in 5 or 10 years, we would have a turnaround time of a vial or of a personalized vaccine vial for the patient, yeah? And the physician gets this vial like he gets any other treatment. So that means the complexity is happening behind the scenes. So on the patient side and on the physician side, there's nothing special. The physician just orders a personalized treatment, yeah, and ensures that the sequence is uploaded, yeah, and then he gets the treatment for the patient, and the patient receives this treatment either alone or in combination. So the question is, what are the costs to make such a personalized vaccine? Yeah? And at the beginning, we had high costs because everything was manual. Yeah? In 10 years, we will have a situation where most of the things will be automated. Yeah? And the amount of mRNA that we would use for vaccines, so we have at the moment, we have cancer vaccines with 50 microgram or 100 microgram of mRNA. So the cost of goods are not the limiting factor. It's really the processing, and the processing must become cheaper by scaling up and by automation.
Yes, and this is also exactly, Adrian, the reason why we chose mRNA, because it's synthetic biology, and it was clear to us even back in the early 2000s that the tailwind which will come from other disciplines, from cloud computing and industry, 4.0 and how manufacturing and automatization will change. That this all will help to ensure that mRNA with its specific features and those technologies which will further evolve will on the one side reduce the turnaround times 4 to 6 weeks, and on the other side also reduce the cost of goods.
Yeah, I think this is a great way to end. I think this is incredibly exciting. I think your passion and vision for precision medicine and personalized therapy is exactly how I feel. I mean, in part, some in the audience already do this. You know, we have sequencing and people are developing these personalized minimal residual disease tests based on ctDNA, based upon that sequence now. So we're not measuring a kind of non-specific antigen. We're now looking at the mutations that were in that tumor. And so I want to thank you again for taking the time for this fireside chat. This was really fantastic. And I'm sure everyone in the audience will be clapping and thanking you. Thank you.
Thank you, everyone.
It was
Was a
such
pleasure.
a pleasure.